And we're going to explore lots of patterns, but we want to focus it a lot on morphogenesis, right, on the biological nature of pattern formation. So today I also want to open up a little bit more, having conversations with you guys about some of the concepts that we will be looking at, and giving you some actual examples. Yes. Giving you twice? Yeah? Giving you actual examples of how we go back between simulation and experiment, and how we go from simulation to theory, yeah? So we model lots of things, like some of the stuff that we look at are plants, for example, but I really like doing modeling or applying physics to biology. It's a little bit like painting, right? So we do not just want to take a picture, a photograph. We actually are going to interpret whatever we are looking at. And for example, this is the painting of Mondrian, which is like a really, really interesting abstract painter from sort of Bauhaus painter from the Netherlands. And he was practicing of going from very detailed to somehow getting the essence of the tree, right? And so this can be sometimes very handy, and we want to do this, but then sometimes we also have to go back to, to a little bit of that mess, okay? And so this is something that I want to practice with you in some of the case studies we're looking at, and I hope that you guys will carry away with you as a search image when you do your work. Right? It's not just about making a perfect imitation of a system that you're looking at, but sometimes if you really want to be, surpass a certain level, you will have to be able to toggle between these levels of abstraction, because we get deeper understanding sometimes, although this might be a little bit wrong, so we also sometimes have to go back, okay? So the things we will be exploring today, I'm going to talk about morphogenesis, what I define about that, and we're going to talk about some of the working concepts in this field, because it's an interdisciplinary field, and people from different fields, from genetics, from development, from ecology, from mathematics, from physics, they will use the same terms, but meaning something very different, okay? So I just wanted to talk about a couple of those concepts which go wrong, right? So you guys will be primed, but you can make your list as you continue in your careers about the concepts which are quite different. And I want to talk about bringing in something that we haven't talked about too much, which is evolution. Doesn't mean that we have to know every single step that happened, right? It's not that we're going to do an experiment about evolution in the same way that you wouldn't do an experiment for a Big Bang, right? But you can say things about evolution, and you should take into account the fact that everything we're looking at has been processed through evolutionary workings, right? So as some say, nothing in biology makes sense except in the light of evolution. So if you want to go into biology as a physicist, you have to start thinking a little bit differently as well. And there are ways that we can do that, and there are lots of techniques, lots of simulation techniques, in which we take into account evolutionary processes. We're going to see some of them today. Yeah, and in the future, if you guys want to have a little bit more technical knowledge about that, that's something I'd also like to give you guys. So we're going to look about evolution, obviously the interface with development, with patterns. And then I'm going to give you some very sort of case studies that we can discuss. It's more thought-provoking. And then we're going to go into something called morphogen gradients, right, looking at what morphogen gradients mean for development. And we're going to look at the roots of plants. The next days, we're going to go more into cell polarity and its feedbacks with cell shape. So it's very similar to some things that Erwin has already been talking about, Erwin Frey, but we're going to look at it in some different systems, and also for moving cells. What happens when we have feedbacks between the movement of cells and the cellular polarities that are happening underneath? And then once you have cell polarity, how do you go to tissue polarity? How do cells couple with one another, their polarities? So that's going to be a theme of tomorrow. And we will talk there's going to be also like a lecture for the institute, so we will talk more about cell and tissue polarity here. The next day, we're going to talk more about the biophysics of the cell. So if you really do want to model these movements of cells, cell changes, what are the simulation techniques you can use to do that? There's a broad range. And then how do we integrate the biophysics of the cell with reaction diffusion? Yeah? How can we do that in a way which is doable and gives you an access to ask interesting questions? And finally, I'm going to give an example which is actually not my work. I will show some of my work, but this is a work which was done a while ago by Stan Murray. And it's one of the works where he goes from single cells with very simple rules all the way to the whole life cycle of an organism. And so what we need to do there is we have to put together patterns like reaction diffusion, chemotaxis, excitable media, spirals, together with a little bit of biophysics, cell adhesion, right? And emergent properties will ensue that can actually lead to the explanation of how an organism is formed. Okay? So that would be sort of, our aim for the week. You guys okay? Yeah? Good. Okay. So that's what to expect. I don't know what you're going to get, but there we go. So it's a little bit of lag here. Ta-da. Okay. So again, my focus when I think about patterning, mostly in my lab, it's not on the eco scale. It's really on the scale of development. And it's not only on the scale of biochemical patterns, but it's on the form. So that's what we call morphogenesis. Now, when we do this, there's a lot of things we're doing. So this is actually really funny. There's like the meta level of how we do science when we do something so complicated, right? So this is a really cool photograph. I don't know who took it, but he actually took the picture here of the guy doing this thing. So there's a lot of things that we sort of abstract and envision. Now, one thing that I realized, I just want to make sure that we're on because we're going to go a little bit more into the biology. Does anybody know what the central dogma of biochemistry is, of biology? Yeah? Okay. Okay. For those that don't know, it's very simple. It's the fact that we have information in the DNA, right? The DNA can duplicate itself. Things that duplicate itself will make errors. There's a mistake, a rate of mistakes that it does. In fact, the DNA we have is way above the information threshold that we could carry. We have a bunch of repairing stuff that makes sure that the errors aren't propagated too much. It can also then form RNA, and through that messenger RNA, this will be translated. So this is called transcription, DNA to RNA. And from the RNA to basically, you have different types of RNA. You have a whole zoo here. But from here, you can make your proteins. Use these ribones and you make your proteins. Is that okay? Yeah, okay. So what is it that we give to our offspring? We give the DNA, right? Yeah. Actually, we give a little bit more. We give a mitochondria. At least the girls give a mitochondria. The guys not. We give a cell. There will be spatial information there too, right? But when you think about inheritance, you're thinking about the DNA. Yeah. Now we also give other things. We give like little epigenetic modifications on your DNA will also be given through. So if someone here during pregnancy suddenly tries to starve herself, your child will have a propensity to become obese, and so will that child. Yeah, so it goes through. And who uses this a lot are plants. Because if I grow a plant here in Brazil, after a while it'll know when seasons are doing, how seasons are shifted. A plant that I grow in Sweden will have to change totally its sense of when the winter and the summer are coming. Okay, so that's epigenetics. So there's a lot of type of cross-communication here. So a lot of biology is taking this here and saying, hey, it's a little bit more complicated, we have a feedback back and so on, blah, blah, blah. But basically, this is it. Now, this is the problem, right? Because at a certain moment, biologists got the whole DNA of a whole human, and they were able to piece it together like a book. So they got little snippets, and they were able to make the book. Then they were even able to take it from a person and personalize the DNA sequence. And because it was such a feat, right, such a technical feat at the time, they thought that they would understand disease. Yeah? It's all there because it's all written. Isn't it all there in that code, right? And this is one of the issues that if you enter biology, and also you should arm yourself with this, the knowledge that there's more to it, right? I'm just going to give you an example. So James Watson, anybody heard of him? Yeah? Found out DNA? Okay, so when they did the Human Genome Project, big thing, lots of money, he's like, I want my DNA to be sequenced. The first guy got his whole DNA sequenced, right? And he's a carrier, homozygous carrier, not heterozygous, homozygous. He has both of the genes of two syndromes, the cocaine syndrome. It's a mutation in DNA repair enzyme, ERCC6 in this position here. So what do homozygous people have? They will be characterized by growth failure, impaired neural development, premature aging, and sensitivity to sunlight. Well, I think he might have some sensitivity to sunlight, but given that he won the Nobel Prize, and he's still alive, he's 96, he really doesn't have that as a phenotype. But his genotype does, yeah? He also is carrier, look, he's really lucky, of the Usher syndrome. So here, this would predict, if you would just look at that he would be deaf, and he would have lost his vision. He's actually not deaf, and he still has his vision. So he's okay, actually. And this was one of the surprises, and people don't tell that to you too strongly, because, where would we be with biomedicine? But genes act differentially, right, depending on other expressions of genes, but also depending on, for example, the physiology of the cell and other conditions. So it's more like a landscape that you're forming. It's not that deterministic, at least not always. Okay, sometimes it's a different story. So with this, it's an issue because nowadays, a lot of things like you'll have this, welcome to you. They'll say, we can get your DNA. We'll tell you everything about who you are, right? And there's a lot of you, which is in you, in fact, right? So for example, if I want to test someone here and say, how good would this drug be individually for one of you, those tests based on your genome are not going to give me any type of significance. What will give you more significance is what are your bacteria in your gut doing. And sorry, it's not you, the bacteria in your gut. It has a different genome. It's not even, right? Not even cross-talking. So lifestyle also, not the genome. They predict, for example, cardiovascular diseases. Although there might be some inheritance, most of it will be about, what you do. And then, it gets even worse when people want to try to identify genes like intelligence or these higher level type of, genes. Okay, so basic story, there's a lot of development, right? There's a lot of things happening after that, that DNA to RNA story. Okay, so even if you want to go into looking at health, at the end of the day, you're stuck with a problem of ecosystems. Because, for example, more than 50% of your cells will be prokaryotic. There will probably be about 160 species in you. And if you take a Amerindian in the Amazon forest, he'll have much healthier guts. Has much more species living there, which is really good, okay? And these things, they actually are known to, change the way we develop and change also the way we think, change the way we process stuff. Okay, so approaches that we need, obviously, we won't be able just to do this mapping from the gene to a phenotype. Phenotype, I mean, by some characteristics that you are expressing. But you'll have to look at these systems, right? Be them systems of cell division, how is that working? Be them metabolic networks. Now, we're not talking about metabolism in this workshop, thank God, because it has its own way of calculating and doing your calculations to make sure everything is right. And what you guys already saw, you saw some simple ecological systems, yeah? But these systems can get quite hairy, right? Okay, but now if we jump also into the development, the morphogenesis of the story, which is what I want to talk about, we will also have to think about, these systems. So it's not just about, what genes do we have here, but for example, even if and it's not even about the network I have, yeah? Because this little network here, these two genes, they mutually inhibit each other. The blue gene inhibits green, and the red gene inhibits blue. Depending on the parameters, it can give me a stable limit cycle. So the topology, same topology, which is, for example, important for somatogenesis, which would be the forming of the somites of your back. It could give you a bistable switch. Either the cell is in the green state or it's in the red state, depending on some inputs. Or it could give you damped oscillations. So there are different type of things, which is very important for neuronal differentiation. Okay, But now also, if we start looking at patterns, and this is what we're going to talk about today also a little bit, we have the formation of spatial patterns which give positional information to cell along an organ, so they know what to do. And even if you don't look at things like hormones or switches and just look at cell sorting, you're going to see that you can get pattern formation just by the adhesive properties of the membrane of cells. Yeah? So all of these things are ensuing without it having to be written, the answer, in the DNA, right? Okay. So you can start seeing how sensitive this issue and this system will be. So, still part of my intro. The problem here is that we have a pattern really beautiful in front of us, something like this tree of life carpet. But when we think about the components that make it and how it's wired, we look at the back of the carpet, yeah? And it's very difficult to get a correct mapping here, right? Especially because when we do some type of these studies, we always look at a very small part of the carpet. And this is what we call the genotype to phenotype space mapping. Have you guys heard about this? Yeah, some not? Good. So basically, genotype is here. It's that stuff that it's the story that you inherit blindly. And we want to predict, okay, how does this bring you here to, I don't know, being faster or being taller or whatever it is. Yeah, some type of phenotype space. So a dot here will take you there. What we see is sometimes a dot here can take you to 2 points, right? So it's not a one-to-one mapping, and it's also not exactly the interesting thing is we know that sometimes you can have lots and lots and lots of mutations, nothing's happening. Another moment, you have just a teeny little, 1 amino acid difference, bang, you know? Not even viable. You fall off the cliff, right? So you don't even develop. So there's a lot of things going on here about how the genotype space goes to the phenotype space, and this is what we really are thinking about when we think about development in biology. We're not in an ergodic space where everything is possible. No, there is some structure here. Yeah? And this is a structure we know we won't ever be able to really know what it is, but we have to work around that. Yeah? Or guess certain properties. So how did that structure come to be? Right? Was it just random, like, poof? Well, obviously, development shapes it. There are certain things which are possible developmentally. Certain developmental processes are robust towards differences in, certain kinetic rates or certain diffusion constants. So development is something which will provide plasticity and robustness and will provide mechanisms which can be reused and reused. So understanding mechanisms of development as a physicist can, you will be understanding that part of the shaping of the genotype to phenotype mapping. Is this sort of clear? Yeah. The other thing is, well, what is going to be selected for? I mean, development is a thing which gives you the constraints and the possibilities of innovation. But what will effectively be on the table is what survives. And what survives is what survives better than anything else which is surviving at that moment, right? So we also have then the fitness and the ecology. So we have to sometimes also imagine that, we might have gotten a very different selection if we didn't have a certain ecological situation, okay? And obviously, all of this is happening on different time scales and on the evolutionary time scale, right? So that's where evolution also comes into play. Okay, so that's sort of the idea here. And you can start seeing how difficult it is to sort of navigate these type of problems. So everybody tries their best. People look at different systems. People from different fields are working on it. And this is what I'm trying to say. It sometimes becomes very difficult to get together and talk and understand one another. So I think Benedict, by now, he has been publishing this book. Benedict Hallgrimsson, he's from the US. And he's a developmental biologist in this area called EVO-DEVO. And he thinks a lot about this, right? And so he has this idea that, well, you have, generation variation by developmental systems within populations. And these are certain terms that you'll see coming often forward, right? And every field will have theirs. But then you have the abstract features of developmental systems in phylogenetic context. But what he says is that fields will have an overlap. There will be an overlap of terms that we use or that we have some type of similar sense of. And that's what he calls the trading zone. That's where we can actually communicate with one another and actually grow, right? Learn something. So one of those words which were up there was called canalization. So has anybody heard about canalization? Cool. No? Okay, awesome. So I mean, I just want to make sure I'm not boring everybody. Yeah? It's good? Okay. So canalization was this really powerful idea from Waddington. And so Waddington is like, you know, there's a whole school of evolutionary developmentalists. And if you read them, they're really beautiful. Because basically, they're thinking like a physicist. I shouldn't say that. I'm being recorded, really bad. But it's about the theory of how these, you know, how these patterns would be coming. But the idea is that you have a ball rolling down this hill. So basically, like, this is a bifurcation parameter. You're rolling down because that's how time is going. And you're going to go into different attractors here, right, into these different holes. So one way of thinking this is development by itself is this dynamical trajectory. In the beginning, all cells are more or less similar. It's not that they know, oh, I'm going to become the heart, I'm going to become the liver, I'm going to become this. But as the things go, you know, they will take those trajectories, sometimes in the beginning for very small reasons, and afterwards they get canalized. So as, for example, a stem cell is differentiating, it can't go back. Yeah? So that's the concept. So in that context, it's called like the epigenetic landscape. So it's the thing that happens above the genes. You know, even if every ball would be the same, it might end up in different places. Yeah. So you could think about this as the developmental phase space. That's the way we would talk about it. And his idea was that evolution also selects trajectories like this. So certain trajectories will be canalized, and so that's the canalization theory. So that means that evolution will have a tendency to make sure that you go certain trajectories and you'll be robust to perturbation. So it is amazing how much stochasticity are in these developmental embryos and stuff like this, but it usually goes right. It's such a complicated feat, but it usually works really well, right? I mean, for the problem it's trying to do, it's quite wonderful. So that would be, you could start understanding it with this. Okay. So, stem cell differentiation, it can't be reverted. Now, I just want to make a side note that if you go into a community which is working with plants, they will have no idea what canalization means. They'll think you're talking about this. Okay? I just wanted to put it up. So canalization here is what you guys, I think one of the groups also had a project on it. You know, when you were thinking about how do the streams form, right? The erosion in the sand. It's very similar. So the idea is if you have growing meristems of plants, you're going to have these veins that start forming. It's an auto-organizatorial process. And what's happening here is you have pumps which are bringing a hormone forward. So they think that if you have any system where the flux will feed back on its own flux properties, so you're enhancing the flux by having a little bit more flux, you will get canalization. So that's another way that it comes up in development. Okay. So still on the concepts, growth. Now, you would think that growth is a very neutral term, right? And so people have massive discussions on growth. So we're going to talk about, yeah? So growth, for some biologists, will mean if I have a cell, yeah, and you think I have like little I don't know, if I have a cell like this, and later my cell is bigger, right? It has grown, because it's just like bigger like this, yeah? So if I have a tissue, and I put little specks on it, and I see that these specks have, you know, come apart, like strain, that would be growth. Now, for others, growth means how many divisions have you gone through. They will be talking about growth as division, and they actually are quite not worrying about the size of the cell, yeah? Now, if you are thinking about mechanics, just taking something, a rubber, and stretching it, is that growing? No, you're deforming it, right? But you're not accreting mass, so that's not growth. Okay, so that's also not the definition of growth. And in fact, you have also people which are very much concerned about the carbon intake, the physiology of cells. They would say if a cell is growing just because its vacuole is getting bigger, but it's actually not acquiring more like carbon. It's not growing. Okay, so there we go. Just be really careful that whatever field you enter, you have to be ever so careful about what you mean by words, even if they're so simple as growth, okay? So these are some of the concepts which are the high contention. Obviously, models we've heard a lot. Emergence, right, in the eye of the beholder. When you understand something so well enough that it doesn't surprise you, it doesn't emerge, right? But there might be ways of defining this where you say, but I let the rules operate on a lower level, and I don't control what's coming out, right? So I don't know. Multi-level is also one of those words which people have long discussions about. Robustness, plasticity. This is something which is waved around a lot. You know, so in biology, just realize if someone says something is robust, it doesn't mean anything. It's always robust to A, but it's going to be plastic to something else, right? There will be some place that it's going to give, right? So you just have to be very careful of that. Homeostasis, right? So that you have something which is changing and you're able to keep that parameter or some internal variable at equilibrium, despite of that. Now, there are lots of discussions here if this has to be through active feedbacks or not. And patterns. So patterns is something that is actually quite contentious. So I just wanted to ask you guys, do you have a good definition of patterns? Or patterns in biology? What do you think distinguishes a pattern in biology from like a normal pattern?
Anyone feeling brave?
Discussion. I'm just talking a lot here, guys. So no? Yes.
I mean, something that you can see that repeats, and like a small unit that repeats, and you can see like the river example. You see branching, and if you zoom in, you can see like the same thing.
Mm-hmm. So your nose is not a pattern, because you don't have multiple noses.
I don't know. Well, other people do. So it's a pattern in
Some
the
people have
population.
Yeah, it's good. Okay, so I agree. So sometimes patterns are thought about as something which is tessellating or repeating or fractile or self-referential and stuff like this. Okay. So yeah.
We have one more.
Very good. Thank you. Thank you for your answer. It's great.
I don't have a complete answer, but I know that, for instance, in comp science, you basically have an underlying grammar, which is basically, if you have a system that arises from that underlying grammar, that is a formation of a pattern. So, and I think we are trying to do things slightly backwards in that same sense. We are saying that something is a pattern and then look for underlying rules that might form the pattern, but I think that is...
Yeah, no, that's also a good thing. So there's a part of morphogenesis, like Lindemeyer systems, where they think about algebras. So for example, if you look at a branching tree, right, you'll think about, okay, I have an iteration, it's going to make a branch, and you can write rules of branching and angles and stuff like this, and then you can apply them self-referentially, and you can get branching structures and patterns. So they can classify mosses and plants and phyllotaxis with those algebras. That's true. But again, I think what we're hearing here is that you guys are thinking about patterns as sort of repetitive units, modular structures of something, right? Could be. I hope that I'll give you another way of looking at patterns. I won't define it as yet, more because I think it's really difficult. But I'll show you a pattern when I see it and why I think it's a pattern. Okay? So we're going to do a little bit of some simulations, some practicals also during this thing. So these are the sort of, I always tell it to my students when they're learning how to model, this is sort of your like toolkit when you do simulations. So ordinary differential equations that we talked a lot about. We didn't look too much at finite state machines, but you know, when we're looking at biology, right? So usually we're thinking about time, we're having some variables, and we're having space. So I just keep it simple like that, right? Obviously, you know, you could do this different. You could be looking at partial differential equations in a phenotype space, right? I agree. But just for the patterns that we want to look at, this would be the way that you could envision it. And one thing you have to just realize is like, okay, is my variables discrete or not? And you heard a lot about the importance of being discrete, and we're going to see a little bit more about that. Is my time continuous or discrete? So when we talk about maps, for example, one generation, something happens, and then it sheds seeds to the next. Be very different dynamics, as you know, from the logistic curve, than if your time is continuous. Partial differential equations we will have a look at, because we are for cell polarity. But I also wanted to explore a little bit like cellular automata and agent-based modeling. So, okay, for the cellular automata, you guys have all seen Game of Life? Yeah. Why do you think it's called Game of Life? Do you think it was just because it was, yeah?
It's a metaphor about like it's an optimal density of population. If there's too high density, then you run out of resources. If there's too low population, there's no reproduction. Like it's like it's a very sort of crude metaphor for what it's doing.
Okay. Oh, you're saying that you could think about population dynamics even with
Well,
that.
I mean, so it's not really a real. It wasn't built to model population dynamics, but it's like sort of population dynamics is sort of a very rough analogy for what it's doing.
Yeah. That's true. But you know, you have other models where you don't see these structures, these spatial structures that give you like sort of densities. But we don't call them Game of Life. We wouldn't. Yeah. Yeah, exactly. Because of the open-endedness, and because also, I would say one way of looking at this open-endedness is, okay, you can read here afterwards all of the things people have said about cellular automata, right? You know, so you only have a state, which is 1 or 0. You look at your immediate neighbors, you count up your neighbors, and you make a decision what you're going to be the next time step, right? So you're either 0, 1, so very discrete. Time steps are discrete, yeah? And the rules are ridiculous. So any live cell, so occupied lattice point, with fewer than 2 live neighbors dies. As if by loneliness. He actually wrote that. It's so cute, okay? Any live cell with more than 3 live neighbors, so if you are a live cell and you have 3 live neighbors, including yourself, it will be overcrowded, so you will die. And any live cell with 2 or 3 live neighbors stays alive. Okay, so there we go. So you have these rules of how do you turn on and off, right? And then if you're a dead cell, so if you're white, but you happen to have 3 live neighbors, you come to life. So here, like reproduction in this little world, you need 3 parents. That's quite cute. Okay, then you go. So it's just sort of random rules. These rules do mean something in their type of complexity of rule table. Yeah, so I'm not going to get into that. But that puts you in this edge of, yeah, edge where interesting things ensue. But the nice thing is that if you start with initial condition, you know, you will see this pattern propagating. Did you see this thing just going away? By the way, this is they made this for John Conway when he died. I thought it was very beautiful, like, you know. But so, this is a glider, yeah? So you have these structures that emerge. And if you play enough with them, you actually quite like them. No, display. Second. Keynote quit unexpectedly. Okay, just a second. Reopen. So what I want to say is, although we have these stupid rules, you start having fishes, and when they collide, they can make different particles. And you have ways of making those particles. You can have glider guns. So they would just make like frequencies, they will make those gliders. And you guys know this, right? And then you can make industries of glider guns. And you can go to very complicated levels. So it starts becoming better, if you really want to do these crazy things, to talk about the mesoscopic emergent properties of that rule table and not think about the rules. Okay? So you start seeing something else appearing. But obviously, they don't do anything by themselves. It's just the rules. Although you can start describing interactions and properties, and you can compute lots of things using these other levels. And that's one of the things, although this has nothing to do with life, which is just to make you think that it can be possible, in very easy terms, to have something which is programmed in a certain way, but where the usefulness, or when we look at things, or we look at the patterns, you describe it on another level. Okay? So you actually have almost unhinged yourself from how it's actually working. So you can also make different games using Game of Life. You can make voting rule using Game of Life. You can program anything using Game of Life. So you're doing something else computationally. But the computations are still that, do I have 3 neighbors, 2 neighbors, 1 neighbor, right? And that's the part which is really important to grasp, to grok. Because a lot of biology will be like this. When people say, oh, didn't you take into account the chemical this or that? Depending on what you're looking at, you might be above that coupling, okay? Now, guys, you can disagree with me on this, because this is a little bit my opinion, yeah? This is not like, so this is not a truth. This is just like the way I look at it. Just a second. Okay, I have to go to the next directory. Open. Okay. Play. Is something good. Okay. So maybe didn't like the Game of Life. Okay, so yeah, so you see this is a blinker, right? And you can understand it. This is a fish. This is these guns that I was telling you about. These are a bunch of factories of glider guns making glider guns down here, right? And yeah, okay. So I'm gonna jump a bit of this, because I'll show you. Okay, so now, still thinking about these concepts. When we talked about when Alvin talked about the rock paper scissors story, why do you think you talked about it? What was important for you guys from that? Was it that there was a diffusion length scale and it stopped? What did you think was so surprising? Or was it not surprising? So I want to show you guys something, if this is possible. I might need some help here from someone with a display. So I need to go here, just like display setting. And main display. Extended, it doesn't mirror, right? No, mirror. I want to mirror now. Okay. So you see this now, yeah? You see my whole mess of my, yeah? So let me just here. I'm just going to quickly show you a little simulation of rock, paper, scissors, okay? We'll do it together. Net logo. I'm showing you net logo because it's something which you guys could do. Is there a way of making this bigger on the screen? Okay, you guys sort of see it? Okay, so we set up. It's going to have those three species, right? And you have that cycle that one eats the other A, B, C, right? And go back to A. Now here we have it a little bit probabilistically, right? So we have it on a CA, yeah? So, sorry, this first thing here, it says swap rate exponent. So that is saying if you have 2 neighbors which are occupied, how quickly you can, like, what's the probability of you swapping? So the bigger that is, the more diffusion you have in your system. You have reproduction rate exponent. So that is, if you have a black space next to you which is not occupied, what's the probability of you occupying it through reproduction? Okay? And then you have something like a select rate exponent, which is the probability of you selecting your neighbor to play the game. And if you play the game, you win, you occupy that position. So you can play with those things, yeah? So if we just go, yeah, I'm going to pause. So what you see is you start seeing this whole population is moving around, doing its thing. And here you see the concentration of everything. By the way, I didn't tell you, I'm doing something a little bit naughty. I am reshuffling at each instant, after each thing, I reshuffle all positions to a random place. Okay? Why would I do that? Why would I reshuffle? So if I work with a system of ODEs, yeah, and I know that they're going to diffuse now, but the first thing I can do is I can put this on a CA, I can reshuffle to make sure I get exactly the same dynamics as my ordinary differential, my reaction part of the equation. Yeah? So the trick to do that is you implement your simulation, yeah? And then you just take this one, you take that one. After you performed all the rules, you just, randomly switch things far away. I mean, far away. It's just going to do random, right? So you're just shuffling. It's called well mix. This is the well mix case. Now, what was interesting about the rock, paper, scissors is that if you look in the well mix case, what do you think will happen? Yeah, it'll go to one attractor. And if you would calculate that out, you would have 3 attractors. It'll just pick randomly, like every time you. Okay, so for example, who do you think is going to win now? Do you see the green curve, the blue curve, the red curve? Who do you think will win? Red? Okay. Yeah, good. Okay, so now I start it again, right? And we can do it like it's nice, this program, because it uses a different random seed. So every time you'll see someone else. You think it will be red. Okay, so this is the fun thing. First it becomes really blue, and then you think blue has won, but it actually is outcompeted red. And so then you just need to have non-zero green in it, and it will take over. Okay, now this is why rock, paper, scissors spatially is so surprising. Because if we just think about the game, well mixed, one of the things is that all 3 are equilibriums. You go to any 3 of it. So then you can put your unshuffle thing on, yeah, set up, and now you're going to start getting the coexistence, right? Now the coexistence only works because there is pattern. Yeah? It's not that the pattern is something to the side. No, the pattern is what makes the species coexist. So for me, this is a pattern because it has a biological function for the coexistence, and I will get there, it's probably been selected for, right? It is a mechanism. It is a biological mechanism. You wouldn't be able to explain through the reaction rate, through the interaction, to the topology of that network, why those 3 bacteria or enzymes would be able to coexist. Yeah? Okay. And then you can do all these cool stuff that Evan has been talking about. So you could put the diffusion up, right? Then you're going to see these patterns smudge out. You could put the reproduction rate up if you don't want the black spaces. You can get them moving quicker. So you can explore whatever you want here. Okay? So this is how NetLogo works. It's quite nice to explore things. Once you want to do serious calculations on larger scales, I would always advise to code this into something else and run it in your cluster, right? But it's very nice for exploration and to try different rules. And it's also very nice to shuffle things around, make sure that your implementation is exactly like what your ordinary differential equation was, or your partial differential equation was without the diffusion, and then you can put your diffusion back in. Okay? And then, of course, it's not going to give you the same thing as your partial differential equation because we have a discrete system with discrete spatial scales and so forth, yeah? But you can at least then compare what the discreteness is doing on top of your PDE. Does this make sense, like as a way to do simulations? Okay. So I'm going to close this. And then I'm going to, sorry guys, just have to, what did I do here? Have to go back to my display. Now I'm going to go. Yeah, okay. So now I'm going to talk about this work, which is rock, paper, scissors. Basically, it's by Maarten Boerlijs, and you can look up his papers and other things. So this has, it's not totally not my work, but I really love this work. So he was looking at epidemics. So where you have a SIR model. You guys ever heard about SIR models? Okay. And so he actually implemented this SIR model in an agent-based manner. So what is a SIR model? So you have an infected lattice site, and you have a susceptible next to it. So there'll be a rate beta, right? Probability here that it's going to infect that. Yeah? Then there's going to be a time scale tau 1, so that'll also be this, this random probability of it to be recovered. Yeah, and then from recovered to susceptible. So when you're recovered, you still have immunity, so you can't be infected again, and you're also not infecting, and then from here you go to susceptible. So you see that you have these sort of category shifts. And again, this one will be invaded by I, and so we go. So we have these transitions of the states. Okay? This looks familiar, right? Yeah? Good. Okay. So now he did something funny. He said, okay, he started exploring differences with these parameters, and he saw that you could get different things. You could get coexistence, or you could get the thing spreading and going away. But when you started getting coexistence, you had, he would call flow regime, turbulent regime, and so forth. It's the way that these patterns move around. Okay? And they have to do with the combination of these rates. Yeah? And what happens is that you will get different sort of patterns. And what you have is very similar type of wavefront movements and these spirals, these mesoscopic spirals that continue to be there, to be persistent. So now he said the following, okay, let's let two different diseases into our simulation, and we let them mutate. They're just evolving. Yeah? So they'll be sort of competing with each other because they're competing for space, yeah? And we just let those parameters evolve. Now, remember, if this were one species, you would get, and you would want the disease to, or a well-mixed system, you would probably, you get an increase in the R0 value and how quickly you infect. Yeah. But if you have this evolution, what you get, and so this is the parameter here, infection periods and infection rate. So these are different simulations, and they all end up in this sort of magical line, which is a trade-off of how quickly you infect and of how quickly you infect and this infection period. And so it was really interesting. So you're not evolving to be the most spreadable disease, to have the highest R0, not at all. Okay. And why is that the case? The case is here. So if you have two cities, he calls this the tale of two cities. So he has one infection here and one infection there. What you see is, so here, this purple color, this is your disease coming. Behind it are the people that have just recovered, which have some type of immunity. Yeah? They will also have a lesser immunity, but also a level of immunity to the other disease, which has mutated. So what happens, like in excitable media, when these waves collide, they annihilate each other. Yeah? So it is the dynamics which has quicker periodicities, which is the one which is able to invade spatially the domain of the one which has slower periodicities. Although the R0 could have been higher, but it's the frequency as which you make these periodicities which will determine who will take over evolutionarily. Does this sort of make sense? You see, so this is in a certain time. Now look what's going to happen. This wave is going to meet here. This is going to die. The next wave is coming close. So it's inching its way into the territory of 2. Okay. So the reason that we get this type of evolutionary tendency is, again, through the patterns. Yeah? So you let it evolve. You will show that you have a certain trade-off between the way that the disease will evolve, and you're not maximizing R0. Now, this is super important to understand disease, because we also think, oh, if a disease really wanted to do its best, it could just, be really crazily bad. It's not in the interest of a disease. It's not in the interest of a disease if it has quasi-species, if it has mutants and variations of itself, to be that aggressive, because it actually it's wanting to stay alive, but it does that through spatial patterns. Okay? So this is a clear example of why it's important to look at evolutionary contexts or competitions, right, where we see that the space will really determine what's going to come out. You can't just apply a random like fitness on top of your equations. Is this message sort of there? Okay. So I have a proposal for you guys, and I had this. Do you guys hear?
What the fuck is that? Rock, paper, scissors, lizard, Spock was created by internet pioneer Sam Cass as an improvement on the classic game rock, paper, scissors. All hail Sam Cass. Hail. How does it work? Oh, it's very simple. Scissors cuts paper, paper covers rock. Rock crushes lizard, lizard poisons Spock. Spock smashes scissors, scissors decapitates lizard. Lizard eats paper, paper disproves Spock. Spock vaporizes rock, and as it always has, rock crushes scissors. I'm sorry, can you repeat that?
Okay, he repeats it. Yeah.
Of course.
Because he's Sheldon, right? Okay, but this is it. So, so scissors cuts paper, paper covers rock. Rock crushes lizard, lizard poisons Spock, Spock smashes scissors, scissors decapitates lizard, lizard eats paper, paper disproves Spock, which is really funny. Spock vaporizes rock, and as it always has, rock crushes scissors. Okay, so now what you see here, not only do you have 5 species, but you see inside, you have these really nice symmetries. So we have from here to here to here to here to here to here. Yeah, so we have these cross cycles inside. Okay, so although this feels a little bit like science fiction, what relevance does this have to biology? Well, this is the thing, this is the abstract tree, right? When we think about like prebiotic evolution or enzymatic reactions, we tend to have a lot of guys and components interacting with one another. So the question is, what would happen spatially to this? And you guys could just take the NetLogo program if you want to and extend it to this system, because I don't think we really know. And what you could do then is you could also let it evolve to see, because one thing which is sort of clear already is that your spatial scale will be much more thin. It's going to be more difficult to fit. So you might have to have some adjustment of those parameters. And that could be done evolutionarily, right? So this is a possible little project. Okay. So I want to give you another example where we cross all scales. And this will be the last thing about ecology I'm going to talk about. And this is we call eco-evo-devo. Okay, because it has ecology, development, and it'll have a bit of evolution. And you're going to be like, how the hell is this? Okay. So these are fields in Wisconsin, United States. And, farmerland and so forth. And there's a disease called aster yellow. So this is a disease which is a bacteria which enters plants, and it modifies the growth of the plants, and it's terrible for farmers, right? It'll make your carrots like really puny and taste bad. It makes sure that your fruit trees don't give any fruits. It destroys basically your yield totally, yeah? And it's propagated by an insect. I'm going to tell you soon. Because of that, they have something like 20 years of data collection, because it's a really bad pest, yeah? And as climate is warming up, it's going all the way into Canada, where it shouldn't be. So we have like data collection with satellites, with people in the farms. This work was led by Stan, and these were the people involved. And Saskia Hokenhout, she is someone who understands a lot about insects and plants. So you can put like an infected plant, right? You have a little insect here, it's really cute. And you have a non-infected plant. And you ask, oh, where's the insect going to go? Is it going to go here into the infected, or is it going to go the uninfected, right? So you can study how attractive it is. What seems is that infected plants, it attracts the insects to come to it. It's like, eat me, yeah? The other thing that the infected plant does, so once a healthy mosquito is here, it's going to get infected, yeah? If an infected mosquito lands on an uninfected plant, it feeds, and it lets its bacteria go into that plant, which modifies it, yeah? Okay, so, I mean, it's just so crazy, the system, because how does it modify? So how does this, so here we have our culprits, right? We have this flying insect. This is like a leafhopper. There are different types. It'll be infected. The infection to the leafhopper doesn't really change its fitness too much, so it's really a host. Yeah? When it feeds, these things will be put into the phloem. It'll enter the current of the plant. And that bacteria, it carries something called effectors. These are certain genes, and there's a variety of these genes. And what do these genes do? They are transcription factors. They go to the plant stem circuitry, those proteins that tell a meristem, now you're going to become a leaf, now you're going to become a flower, and they repress and activate things. So they hack in, it's like a virus, they hack into the code of the plant, because do insects eat flowers? Nah. So they make it really green, really appetizing, right? They make sure that it is very attractive to the insect. So they unleash a series of changes into that plant. And it's really bad for trees, for example, because they're never, ever going to have fruits again, right? So it really destroys their development and that genetic level, yeah? OK, so this is the phytoplasma. Phytoplasma is that thing here. This is an example of an infected periwinkle, so it'll change. So the problem is, the question that we started to ask is, well, first of all, you have different type of effectors. Why doesn't one outcompete the others? This is a question we're still looking at. And the other question is, and we're going to show you now, that if we write, and I'm not going to bore you guys to death, but if we take a look now at these interactions between infected, and the growth rate of the plant, of the healthy and the non-healthy, because, of course, the infected plant will also, it's going to attract a lot of mosquitoes, a lot of leafhoppers, but it will also die at a certain moment, right? If we formulate these things in ordinary differential equations, where these are the host plants, this is uninfected. So here you just have a carrying capacity, and then become infected, right? And here you die, your mortality rate of dying as a plant. Then you have your uninfected leafhoppers, right? Which will become infected if they feed on an infected plant. And you have your infected leafhoppers. So if you start modeling this just with the most basic assumptions you can, what you start seeing is that you should expect quite a lot of damaged plant, around 20%, and a lot of infected leafhoppers. The funny thing is, when people go and they look at the RNA, they actually sequence these leafhoppers, they don't see that huge amount. And also, 20% is pretty much. Like, I know that a farmer might lose a lot or everything, but if you look at a bigger scale, you never would get 20%. So we were at odds, because for any parameter setting, there was no equilibrium with both infected and uninfected leafhoppers. One would always take over. So a well-mixed system doesn't explain it. So zombie plants would take over the world. That's how we called it, right? Because they are zombie because they're like Walking Dead, right? They don't give offspring anymore. They're just there to like spread the disease, yeah? OK, now the funny thing is, when we started looking into the data, you see that when they were looking at these different fields, you'd have it very patchy. Some fields were infected, some were not infected, and so forth. There was some type of very striking spatial thing there. And when you started looking at the abundance and the infectivity, so they'd collect mosquitoes, and they'd see how infective that strain was, and so forth, there didn't seem to be a correlation that the one which is more abundant is the one which is more infectious. This is usually what would happen in these epidemiological problems, yeah? So what we can do is a multi-level model. So this is now me using this word, multi-level, because on one hand, we are describing what the phytoplasma effector genes are doing on the propensity of a plant to attract or to die or to reproduce. We're going to put it on an explicitly spatial representation, right? So we're going to have plants which live on the lattice as concentrations, and we're going to have these leafhopper populations moving around, right? And we want to see if we can get, what we will see there with the result. Now, this is super messy. Like, oh my gosh, what does my space scale mean? How many mosquitoes do I put? Think how messy this is, right? How do you do an individual-based model and are confident that you're not just doing rubbish and inventing rules? Okay, you guys know the answer. You will shuffle it and compare it to your ODE. So this is the results of your ODE and to your IBM. Because now I know that I understand what my parameters and what my calibration of my IBM is when I go to the ODE. I've done all my homework correct. Actually, Melissa has, so it's all okay. So if you reshuffle it, you should get the same well-mixed dynamics. I can't stress this enough. I know this is like, I'm being really pedantic, but you won't imagine how many times people think they found a new mechanism of pattern formation, and it's a bug in the code, or it's a bad way of rescaling your terms. It's really, really important to have something that you can check. Yeah? Okay, so if we reshuffle, as I told you, we'll get that same dynamics. So lots of diseased plants. Your uninfected mosquitoes go down, and you have a lot of infected mosquitoes around. Yeah, zombie take over. But if I allow this to be spatial, right, what you see is you get a coexistence. So now your not infected mosquitoes are actually okay. They didn't get wiped out. They're still very prevalent in your population. Okay? So those same parameters with those rates that they had measured, and they have painfully been measuring, if you put it in a spatial context, it gives you something which is very close, or more qualitatively close to the predictive value. Okay, so yeah. And then when we have the coexistence, we're like, hey, we also get spatial patterns. If we don't get the coexistence, you guess it, we don't get spatial patterns. Is there something there with the spatial patterns, right? So if we make simulations with a low virulence, we get all infected, medium virulence, we have the mixed scenario, and high virulence, infection dies out. And only place we get spatial patterns is the mixed scenario. Okay, does this mean something? So now we're going to do a competition game again. We're going to let these different parameters, these different mosquitoes, these different virulences compete with one another, right? And we did this in two cases. In the well-mixed case, here, and in the well-mixed case, you either get one strain winning or the other strain winning. But your tendency evolutionarily is to get each time more virulent. Your disease just goes out of control, and you have zombies, yeah? Bad news. But here, when you have a spatially structured IBM, you actually go to this ecological equilibrium here, which is where you have these spatial patterns. So you're actually able to go to this coexistence. So what we see is evolutionarily, not only does coexistence mean you need the spatial patterns, but they're being selected for, okay? So the sweet spot is being selected for. Okay, so now we're working a little bit more with them to test some of those hypotheses, yeah? But again, this is where, I mean, what did I just tell you? It's not because it's my work, but this is all the way in biology. We have a gene in I was going to say a bad word, but in a nasty little bacteria. And this is transforming the transcription factors of a meristem of the plant in a way that you have ecological scaled patterns. So what we have to find is we have to find those type of patterns over time as well. It's a little bit complicated because, you know, if you have farmlands, they cut it, but it also goes into the wild plants, so we're looking at how to look at that. And there's some differences with radiation of these plants once they get infected because of the composition of flowers and leaves. So we will try to hope to get something a little bit more, like you don't have to go there one by one and check. Yeah? Okay. Okay, so I have 50 minutes. This is, by the way, the first part of my talk. I have. Okay, so what to expect? Yeah, so this is what I talked about today. I really wanted to talk about Evo-Devo because I think it, for me as a physicist, this was a thing that I really learned new when I started interacting with biologists. Right? There was a whole world, a whole theory, and what I want to tell you guys is like, use it and abuse it, because there's a lot of beautiful things you can do there. Yeah? So patterns are selected for. Developmental mechanisms drive evolution. Okay? This is, I didn't prove this, but this is something we're going to see a little bit more. We didn't talk about morphogen gradients, but I want to tell this story as well, because the story here is that at the moment I'm very hand-waving, but for the morphogen gradients, I actually have the experiments in the lab. So there we actually had to be very quantitative to discover why a certain mechanism was being chosen. So sometimes you can be a little bit qualitative and get to nice insights. Sometimes you have to be, really J-type, look at every single biophysical number that you can to try to understand what's happening. Okay, and just to give you this idea that concepts are like trading zones in interdisciplinarity. So yeah. Now I do have another 15 plus 10 minutes. Should I continue a little bit? Or should I take questions? Or yeah. Second. While you get your microphone, I'm just going to start something up here.
Okay, thanks. Can you go over again how you scaled up the IBM to, at some point, if I understood correctly, there's some kind of meta IBM that you have where there's like different IBMs at different scales interacting, or was that just one big simulation and you were just looking at different abundances in different sort of blocks?
So you have one ginormous lattice, right? And you have, the plants don't move.
Yeah.
So the plants will be like variables which are there and which will be infected and die, and they can also be repopulated. But what is moving around are the insects, and they look on the grid below them, which is those plants. And then all those propensities, like if a plant is infected, they will have a higher propensity to choose that plant, right? There were some differences, like talking about, the talk we just saw. We did levy flights as well, like because we have diffusion, but like, hey, maybe this would work differently with levy flights. It didn't change these type of like the equilibrium that you would get a coexistence. Of course, it changes a little bit, like exactly how those parameters look like, but it's okay. And yeah, and so when we, you have a lot of those interaction probabilities, right? So just on your reaction part. So that's why in the very beginning, because it became very complicated, that we just reshuffled everything. So we didn't allow that diffusion, and then we made sure we had a good match, a perfect match, in fact, with the ordinary differential equation model. And the other thing we did also is we made sure, like, because someone said, oh, probably the results you get, someone said that, like, the result you get in reality, it's just stochasticity. Yeah. So I don't have this here, but we added. It's not stochasticity. You actually get a totally different, behavior. It's not. So we also did these Gillespie simulations. So we have stochasticity on top of it. You don't get. You just get like right. I mean, the equilibrium is just one equilibrium. So you're just going to get like a spread on top of those levels, but you're not going to get a qualitative shift of a different equilibrium. Yeah.
So your intuition for why the spatial model produces such a different outcome, it really is very different from what Nigel was talking about with demographic stochasticity and things like that. It is
in this case,
fundamentally
yes.
about this kind of spreading process where you have whatever, due to initial conditions, you're going to have patches and you're going to have these insects having to first establish themselves and then hop to the next.
I think in this case, yes. Now, okay. Nigel, I have to tell you, has a really cool project that he was talking to us about, like really cool, which would also have evolution in it, to try to show how a predator-prey system can evolve to have a half phase out of place between predator-prey. Again, you can only understand that when you think about something called rapid evolution. So that's something that I think Ricardo will talk about later. Yeah. So yeah. So it's not in conflict with what he showed, but it's a different mechanism. Yeah, I agree. Any other picture? Thanks for the questions, by the way. Really good. Any other? Second one is. Okay, I'm just going to open my, this was still LaTeX, so sorry. Give me a minute. Okay, so now shifting gears, I want to give you a little bit of this idea of what morphogen gradients are. So they're very simple things, but view, oh, now I can do it now. Ah, got it, okay, perfect. Sorry, Okay. So morphogen gradients is a really important concept for developmental biology. So it's, oh, sorry. I have to shift back. So main display extended. Sorry. No. Oh, this is going to take forever. Sorry. I think it's in another. Ricardo, could you give a little help here? Because I can't see it here, and I can't go to the other window as well.
It's frozen?
No, because it's mirrored still.
But the mouse is normal?
Yeah, the mouse is here, but I want to go to the other window, you see? Like,
To which one? You're gonna...
I want to switch I have different screens. So you see what I mean? So I can't see it there and I can't see it here. Sorry.
Because you are sharing a screen with a VGA? Or?
Yeah.
Okay.
Oh, with this one. This is where I'm sharing screen. You're right. I thought it was the HDMI. Ah, perfect. Just a second. So I wanted to show this one. Yeah.
Let's see if it works.
Like this. Yeah. Oh, I have to put this one right there. Now it disappears here.
No.
Yeah, but it's okay. And if I go, no, so I can't see it here. Ah, okay. So I can't see it here, so I'll have to look at my screen. Okay. Yeah. So Louis Wolpert, he proposed this idea in the actually Journal of Theoretical Biology. He was an engineer, a civil engineer from South Africa, and he went to UK. He's a really nice guy. So his idea was, if all cells have the same DNA, how do you get cells differentiating into different tissue types, right? And the question he wanted, so he came with this idea of French flag. Have you guys ever heard French flag model? Yeah? Okay. So how do you get, if you have a row of cells which are all equal, how do you get blue, white, and red? So the idea was like, okay, if you have some type of graded quantity of something, of a substance, like a morphogen, you would be able to say, you know, your thresholds. If you're above this threshold here that you're feeling, you become blue, you trigger the blue gene. If you're in between here, you trigger the white gene. And if you're below here, you trigger the red gene. So it's really, really easy, yeah? But the question that he originally proposed was different, because they had this thing that they had things which were growing, right? So you have a little embryo, which has, like, let's say, these different regions, and it's growing. How do you make sure that it keeps the same French flag? That's why you call it French flag. He didn't want this to become like this, yeah? Like that. So he wanted something that would scale with the system. And he was just thinking about it very theoretically, right? Blue, white, red, we have it bigger, you know, how do we do this? So he went through different mechanisms. And he said, oh, one way to do it, because, you know, he knew a bit of math, is like you could just have a diffusion of your concentration, right? Some degradation, right? And then depending on the boundary conditions, you can have this as a linear, right? Or you could have it as a exponential. So this was it. Nowadays, I will just tell you, this is a big field. So many physicists go work in how do you actually predict your length scales, given that, is it really the case that things are just produced in one location and they're being degraded at a very, you know, at a strictly different place, or is it degraded everywhere constantly? What does diffusion really mean? Are we talking about free diffusion? Because when we talk about development, we have cells, so it might be cytosolic, it might be intercellular. So people are all, you know, discussing these type of things. Decay rate. So not only do you decay in the sense of your chemical becomes unstable and decays, but you could be captured, right? So there's a lot of like different things that could happen that could warp those type of gradients, or might actually not give you the predicted length scale that you're thinking about. Now, one thing that we see is really important, and I'm going to talk about that. The animal community doesn't talk too much about it, but it's directed transport, okay? And I think that a lot of things in animal morphogenesis is probably going directed more than we would like to think. And the reason we don't like to think that much about it is because, you know, hey, these diffusion equations are so nice to work with, right? So simple, okay? So those are just like things for you guys to worry about. And the other thing is about the steady state. So the way that, you know, Wolfram came, it was just, he was just talking about steady state. You know, you grow it and you let the, you know, that morphogen gradient relax to that new growth. But of course, you know, depending on your spatial scale, you will have a certain constraint on your diffusion over your degradation, right? Let me show you here. Yeah? This here, this delta over d, right? We'll define here, you know, your characteristic length of your exponential curve. But that also defines your characteristic time for you to equilibrate there. So sometimes you might want to do your best to fit a spatial scale, but you're forgetting that it would have taken some time for you to get there, and that might not always be sensible, okay? So this is the big sort of intro that I want to give you in things, and I want to talk about plants. So plants, they develop from, this is the embryo of a plant, so they already have a notion what's going to be their top and their bottom parts here. And this is the root, and this will be the cotyledon. So this is a dicot plant. Yeah? But the cool thing about plants is they continue generating new meristems. And so this would be a pattern, right, in your definition, phyllotaxis, because you have like a repetition of those patterns, and you could describe this very easily with the algebra. We will talk about those patterns on another day. But if you look at the roots, they're much more crazy. It doesn't look that beautiful. But what they're doing, basically, they're foraging. So they will have a genotype, which gives them a little bit of like, what type of strategy do you use? But it's very much subjugated on the type of soil, the water availability and minerals that you're in. So how does it work? So you have a primary root. It has a meristem on the tip, and the rest is growing. Later, little different meristems can appear to the side, and it branches. So if you look at a root growing, so this is just like a macroscopic video. What you see is that it looks like it's a pencil that you're shoving down into the soil. But what's happening is you have lots of cell divisions at the tip, yeah? And then as your cells move away, or the tip is moving away from those cells, they expand a lot. So you can see this here. They sort of change the coloring. And it's a really bad quality resolution here. But what you start seeing is getting fuzzy. Do you see this fuzziness? So those epidermal cells, they will make projections. They're called root hairs. So at this moment here, that tissue isn't expanding anymore. So basically what we have, like the way a root grows, is that this here is called the meristem zone, right? So where lots of divisions are taking place, those cells are dividing. Then they stop dividing here. They start expanding. This expansion is basically what's giving it most of its length gain. And then at a certain moment, they stop expanding, and they start forming these little root hairs, okay? So we have these transitions, okay? So like a French flag. We have a meristem zone, elongation zone, and then differentiation zone. Now the question is, how do those cells know that, oh, here I have to divide, 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 and suddenly from one moment to another, I have to stop dividing. Oof, I have to stop expanding. And suddenly here, I have to stop expanding, I have to stay fixed, and I can do other fun things. Yeah? So I have to get my screen back. Oh, no. Yeah, okay. Good. Okay. Yeah, so this is it. So this is a confocal microscope. By the way, your stem cell is right here. Yeah? And so these are the zones. And I'm going to give you a really quick story about this. Okay. So there is something which we know is being moved around the cell. And Darwin had suspected it was there, Darwin and his son, and it's called auxin. So auxin is a very small molecule, and it diffuses very quickly. You heard about auxin? Good, yes. Okay. So the same way we think with our neurons, yeah? Plants, they make decisions, and they think, and they integrate information, I say think, with phytohormones, with these things. because they're able to move it around. And for example, they will know, okay, this is where the sunlight is, this is where gravity is. Oh, there's another meriston coming up, so I better not compete with it. So this is how it integrates developmental decisions over the length of meters, for example, yeah? Through the transport of this hormone. And we have auxin reporters. That means that we can see what cells have high amounts of auxin because they report certain genes. And so where you see the dark blue here is where we have high auxin, so maximum of auxin. Now, I wasn't really sure if they would be high here or not before we did the modeling, but this was sort of the idea, okay. And is this important or not? And how would this, what's happening with auxin and the gradient? Okay, so how does auxin move around? Some people thought, okay, it's produced here because that's where we see it. It should be the source, right? Because that's what Wilpert said. There should be a source of morphogen, and you diffuse, right? And maybe you have a degradation somewhere else. So the idea was like this. Hey, maybe there's something being producing auxin here a lot, and you'd have a gradient extending going down. But they also knew that there was some transport. Yeah? And so these things in green here, they are proteins which stay at the edge of the cell. So if you have a cell, you might have here in a different color, you would have some proteins here, and there would be like pumps, yeah? And they would be able to take the auxin, which is in the cell, and pump it out. Yeah. Now, what's one really different thing about plant cells and animal cells? Like big difference. Yeah, cell wall, right? So when something is pumped out, it doesn't immediately go to the neighboring cell. It'll be here in this in this apoplastic region, we call it, and it can also diffuse around, okay? So we know the diffusion coefficient of auxin was around 800 microns squared per second. We know it's about, so this in the cytosol, we know it's about 15 times lower in the cell wall because it's much more dense, yeah? And then we have these pumps. So where the pumps are pumping things out, we can express these pumps on oocytes of frogs, and we can measure something called the permeability rate, right? So this is not diffusion. This would be the flux, right? Here would be proportional to the permeability rate times the concentration inside the cell. It doesn't care about the concentration outside of the cell. So if there's a certain concentration here in the cytosol, it's going to, this concentration times the permeability rate will be the amount which is leaking out, right? So this is sort of like a permeable boundary condition. So we have diffusion in the cytosol, yeah? This would be just, here your flux is just going with the diffusion constant, right, gradient. And here it's going like this, PC. Sorry, my handwriting, but you'll see it much better up there. Now, the cool thing is that these pumps, they're located polarly. So for example, if we're looking inside the root, in this middle part, all of them are facing downwards, okay? If we're looking at the external cell files, they're upwards. Here, it's sort of all over the place. We didn't really see a clear polarity. And on these boundaries between the lower and the top, we saw a little bit, but we didn't know if it was relevant or not, going inwards into the midline, okay? So basically, the patterns are like this. Yeah? But these are the patterns on the cells, right? So it's not a continuous thing. It's just on every single cell. Okay. So what we decided to do is we decided to take all of these permeability rates. How does auxin permeate into the cell from the cell wall into the membrane? How does it diffuse into the cytosol? How does it diffuse out, et cetera? And we experimentally looked at the pins, at those, and we just put it back into a model. And we made a very simple model of it, okay? So this is how you go from some experimental knowledge to, yeah, and this was our first root ever made. Really, really, really simple. So we generalized the middle part, vasculature. These are the epidermal cells. This is the endodermis, and this is the root cap. Yeah, so these are the characteristic polarities, as we saw and we measured quantitatively from the confocal imaging. Okay? Okay, sorry. These are the equations, yeah? So we have diffusion in the cytosol, diffusion in the cell wall, and permeability along the membrane, but they're very different depending if they have pumps or not. Until now, it's sort of very basic. We're just constructing back in silico roots. Okay? Boundary conditions are always a problem, right? Because you're not going to simulate a whole plant. So what we do here is we just take into consideration the fact that oxygen is coming from the shoots. So there's an influx here, and it's allowed to leak out. So we use a boundary condition as if that root is part of a connected tissue. If there's any questions, so we're going to numerically resolve this, right? So this was a program written in C. They became very big roots, so we do this with alternate direction implicit solvers. Okay, so these are the things. Remember here that the unit of diffusion is micron squared per second and permeability is micron per second. So we can now scale also the resolution of our simulation without changing the true dynamics of what we're seeing, because we actually have a space scale and a time scale, so we can say how quickly things are happening. Okay? Okay, I'm not going to tell you about the numerics. So when you do, this is really bad because I think there was a little thing here. No, my movie. It's not going to show my movie. Okay, so it starts with an empty root, and you will see like you would have seen oxygen coming in, but very, very quickly, and I mean in the matter of 2-3 minutes, this type of tissue segment here already gets a maximum and gets a exponential gradient. Now the funny thing is that this gradient starts moving backwards. So, but this is the quasi-steady state. So this is actually here the concentrations in this cell file here and here. This is the steady state. Here, here, and here. Absolute concentrations, and on a log plot, you see it's exponential. So what we saw is that there was a lot of concentration of oxygen concentrating in what we call the quiescent center cells. Now it's very interesting, because these quiescent center cells, we didn't give them anything different than the red cells. Okay? So it's just because they were at that interface that they accumulate so much oxygen. So they're not producing oxygen in our model. Okay? So this was the first thing that we could tell the biologists, look, those cells at the boundary, they will accumulate oxygen if they're producing oxygen or not. It's just because of the way that the polarity fluxes are located. Yeah? But the reason I tell this story is because people say, oh, okay, the end. You know, you have a simulation, it looks like your experimental picture, and that's it. And so this is actually where your story begins. Yeah? So what was behind that is the fact that you get these flux patterns. So this is like where oxygen is going for every position here on my grid. Right? And this is giving you the directionality of the vectors. And these vectors, the flux, they are normalized because actually the fluxes here at the tip are very, very large. So the throughput through your maximum is the greatest of all. Yeah? So what's happening there, oxygen isn't really accumulating in that sense because it's actually making like these eddies. Yeah? It's moving very quickly around and passing very quickly through your quiescent center. But because everywhere you have some flux going back, right, you end up, you know, having dynamically this, maintaining this high amount at that interface. So we call that the reflex loop. Okay? And the story gets better because if that's true, that what's causing this is the reflex loop, it's how much is going back into your fountain instead of back up, it means that doesn't matter that much how much pins this part of the tissue is positioning down or how much pins it's positioning upwards. What's really important is how much were the lateral pins, how much of that sideways pins was there, because that's giving you the reflex. So we did some simulations here. So this would be like our normal roots, which we call this a certain capacitance. So it has this tendency of accumulating oxygen at the tip, right? If we now would flip the pins outwards, but we kept everything the same, the downward flow and the topward flow the same, at least the pins, we lost the maximum completely, and we have the concentrations being inverted. So we have this type of graph, this type of graph here, red, yeah? And then you could say, oh, but I can compensate that effect by saying that we have all of our flux pointing downwards, and we have nothing going back up. But that doesn't work either. You would just get an accumulation here in your outer cell files, but you would not get a gradient. So in the system, what's making the gradient is not how much is going down or up, but how much is going sideways, this laterality. And this is what was really important working together with biologists, because we want to make mutants of these guys, right? Because we want to control how big our root can grow, our meristem zone. So we want to see which of those pins should we change, and we can change those things, yeah? Should we look at the vascular? Should we look there? And now suddenly it was like, no. Don't look at those guys. Look at the lateral ones. Those are the really important ones which are controlling those spatial scales. Okay, now the other thing I told you is like very quickly, so in like 45 minutes, you get this type of gradient here in this root. This is a cross-section through cells of the vasculature. These little sawtooth are the gradients within the cells, because there's diffusion within the cells, a flux within the cells, yeah? And what you see here is you're going up the meristem, and you have a maximum here at the quiescent center, the stem cell niche. But as time goes by, like very slow time scales, because you keep on having this capacitance, this ability of accumulating oxygen due to that spatial structure of PIDs, your elbow of this, so this slope is the same, but it starts moving backwards. That means it moves away from the root tip, okay? Now what's really interesting is, if we were to suppose, and that was that oxygen is somehow correlated with cell cycle, which it is, you would expect the meristem zone very slowly to expand. And when we look at experimental data, where they're actually looking at where is your meristem zone, they see that a root at day 6 and at day 10, it's growing still. Its meristem zone is growing. It's a very slow process. So on one hand, we get a maximum appearing in 2-3 minutes, but on the other hand, the same system allows you to get very slow dynamics on the order of days, which match the growth rates. And I will finish soon. Just tell you that then we put it in a spatially moving model, where we say, okay, if you have cell division will be correlated with high concentrations of oxygen. So if you have high concentration, you have a bigger probability to divide. Cell expansion will happen more quickly the lower your concentration. And we won't have any threshold per se. So we're going to have a type of, every cell will go through a certain cytokinesis, so a certain cycle of fixed growth. And then it can linger here in fast expansion cycles, the lower the oxygen levels are. And if we put this in a simulation, oh, please tell me this is going to open. Oh, okay. Or maybe it's somewhere else. I don't know what's happening with my computer. I'll have to try to do this afterwards, guys. Okay. But it's quite fun, because you see the root growing. So now we have the interaction between the morphogen gradient and the growth. So in the very beginning, you see here you have small cells, and after 8 days, you have a bigger meristem zone. So this is sort of like emergent property of the system. But what's also really nice is that the threshold between dividing cells and growing cells was sharpened, and that was a reaction of the growth itself. Okay, so this is the mitotic area actually sharpens with time. Due to time, I'm just going to stop here. Well, just 2 more minutes. The model predicted another thing. It predicted that, and I did this once because I had a problem with the boundary condition, that if you would interrupt the flow coming from the boundary condition, what do you think would happen? If there's no oxygen going into your roots, do you think you're going to lose it all at once? So you have a root. You have a source here, right? It's getting some oxygen. It has this reflux loop, right, which is due to its polarity of these transporters. It's able to make a maximum with a very big accumulation at a specific cell here due to its tissue topology. What do you think will happen if I now stop this? Do you think you will lose this immediately? So what happens is, you actually keep the reflux. It's a greedy root, right? This here keeps on circling. So you lose your influx, but there's no problem here. You just keep on recycling that oxygen. It's like totally oblivious. Of course, on a slower time scale, there's a degradation of oxygen. So your total amount of oxygen will diminish. But it diminishes in a way that the last place to have a maximum is your tip. So we did the experiment. So this is a normal root, which is growing. It's attached to the shoot, so it's getting oxygen. It has a meristem zone. So what we were saying is, your quiescent center should stay the same, but your meristem zone should retract, because you're losing, your gradient will be shifting backwards. So we could do those simulations. And that should happen in the order of days. And it's really interesting. It might be a mechanism for the root tip. in case like a cow comes and eats the top of the plant, it's still able to, at that moment, to keep reshuffling and keep its pattern close to the stem cell niche. It doesn't need to be fed continuously by the shoot. Okay, so with that, I just wanted to stop here. So what I would talk later is, we could have a lot of experimental proof why we think this model would be correct. I mean, it is hinged on experiments, and we have predictions that we could follow. But a very important question to ask, so I want to use this as a case study, is to ask, why did the root do it like this? Why does it have to have these pumps and this reflux system? Wouldn't it have been easier just to have a location of production, a breakdown, and a morphogen gradient, as our classical examples coming from Woolpert? And when we start exploring this simulation-wise, we get a lot of good insights for biology as to what it is that the root is actually trying, what are the constraints it's trying to navigate. So we are just really fixated by the idea of a morphogen gradient, but the root is actually trying to do different things. And what it's basically trying to do is trying to grow quickly, maintaining that same pattern. It's trying to be able to reheal if something goes wrong, so if we ablate it. And it's also trying to keep it sort of independence from the shoot in case there's some type of wounding. So those are the sort of the insights that we're able to understand once we start comparing it with other mechanisms. So I think it's time for lunch now. So thanks.
Is there any question?
Yeah. And if there's any biological questions also, anything which wasn't clear, because...
Hi, Veronica.
Hi.
Thank you, very nice. Is it possible for these roots to grow in water?
Yes, so you can grow roots in water, right? The reason I'm saying this, like these roots, they like to grow in, like when you put them in agar, they actually like to go on top of it. They really like a mechanical interface. And then also, depending on how hard your agar is, they like to wave a bit. So in water, I think there'd be like these here might not be too happy, but you could, yeah. Why do you ask? Is there a...
No, I think it could be interesting to see what's the internal dynamics of auxine.
Yeah.
If you have bending roots or stuff like that because of the water.
So another thing is like, a straight root just has this gradient like this, but if the root is tilted, like this is gravity, yeah? And you have the root like this, what's going to happen, it's going to distort its gradient, because it has, here at the tip, it's going to move a little bit more. Let me just think about how this works. It's going to do this. It's going to have more auxin at the bottom, and less here, because it wants to start expanding quicker. So it bends back.
Exactly. Yeah.
Yeah, so it's a really good question, because we can look at gravitropism by measuring how the auxin is being shifted from the left to the right, and then it bends. But then what's really cool, and this is something like people haven't really looked at, once it bends, it overshoots, because now it has a population of smaller cells here which then grow. So it starts actually waving. So it has some type of waviness which has to do with these dynamics of auxin, reshuffling, and the time scale of the cells. And yeah, it's really a beautiful cool area. It's a beautiful area.
Thank you.
Any other question?
Food?
No? Yes? No?
Lunchtime.
Great. Thank you, Veronica, again.
Yeah.
Don't leave the room yet, please. We need 2 more minutes, just 2. So in the list we circulated, there was some people that didn't sign, and I just want to make double sure that it's because they are not around and we are not missing anyone, because that's going to be important later. So I'm going to